Today on the Treat the Cause podcast and YouTube channel, I'm going to be talking about the new theories on the evolution of cancer, whether parasites are actually good for us, plastics and autism, selenium for cancer and the immune system, and 2 new amazing benefits of vitamin D. If you're interested in any of that, keep watching because I'm about to get started. Good afternoon, it's Dr. Greg Emerson on the timestamp 3rd of June 2020 in the beautiful Northern Rivers area of New South Wales, Australia. There's a lot of conflict and pain in the world at the moment. I've been spending my time kind of isolated from it, doing a lot of study, wild food foraging and exploring farmers markets, getting to know a lot of the local farmers. Sitting here as usual with my Native American cup from New York, drinking beautiful coffee mixed with my very special coffee blend, a lot of it made from sun-dried local produce. Be available on my website very soon for people who want to put a 1/4 teaspoon of beautiful local medicinal plants in their coffee, makes it taste nicer. So much pain and suffering in the world at the moment, I don't really want to talk about it. A lot of other people are doing that. I'm just going to focus today on medical research because there's a lot of really interesting medical news at the moment and research, which as usual is continuing to support and validate the use of some traditional health practices in modern medicine. So let's have a look at some of these studies. First one is about cancer being an evolved species. I've always maintained that I think that the evidence that cancer is a mutation in the mitochondria of our cells is now very strong, and that mutation occurs because of exposure to environmental toxins, which is what was documented in the President's report from many years ago. And it's that cell's de-evolution into a more basic life form in order to ensure its survival in that toxic environment. And there's a paper out now in Berkeley Research from the University of California called Are Cancers Newly Evolved Species? And it says, in a paper published in the July issue of the journal Cell Cycle, the authors describe the theory that carcinogenesis, the generation of cancer, is just another form of speciation, the evolution of a new species. As I've said, it's a cell de-evolving into a new life form, a more basic life form to ensure its survival. Quote, cancer is comparable to a bacterial level of complexity, but still autonomous. That is, it doesn't depend on other cells for survival. It doesn't follow orders like other cells in the body, and it can grow where, when, and how it likes. That's what species are all about. The idea that cancer formation is akin to the evolution of new species is not new, with various biologists hinting at it in the late 20th century. Evolutionary biologist Julian S. Huxley wrote in 1956 that, quote, once the neoplastic process has crossed the threshold of autonomy, the resultant tumour can be logically regarded as a new biological species. So that just kind of confirms what many of us have been thinking, that this is the de-evolution of a cell to the toxic environment, and it takes place in the mitochondria. So what I've been saying all along in terms of what I am doing personally to reduce my risk of cancer is to, 1, look after my mitochondria, and 2, address my toxic exposure. And I do that by, 1, minimizing my exposure to toxins, but also having a detoxification strategy where I try and maximize my elimination of those toxins. Because we have detoxification systems in our body. That's our sweat, our stool, our urine, our liver. And many people say those detoxification systems do an adequate job at getting rid of our toxic exposure. But what we forget is those detoxification systems were designed several 100,000 years ago, probably even longer. Let's say longer. Let's say way longer than that. And our exposure to toxins is dramatically increased. We've got systems that weren't designed for the level of toxic exposure that we're now getting. So for me, minimizing my risk of cancer, some of the ways I'm doing that is looking after my mitochondria, minimizing my exposure to environmental toxins, and having a detoxification strategy as well. Selenium. I've written quite a bit and talked about previous videos and podcasts about the role of selenium in preventing viral infections. This has just come out this week in the journal Medical Hypothesis about selenium and viruses. And I've always said that the evidence is very clear that selenium works by stopping the virus attaching to the cell, sticking its hypodermic needle into our cells, and injecting its DNA or RNA into us. The whole idea about a virus is it's pretty useless until it gets into our cells. And when it gets into our cells, it wants to stick that needle in, inject this DNA or RNA, and then it hijacks our ribosomes, which are our intracellular factory for reproduction of cell organelles. It hijacks them to reproduce itself, and then when there's enough of it, it eventually bursts out of our cell, and that's when our immune system reacts against those viruses bursting out of cells. So what does this paper say about selenium? It says sodium selenite, but not selenate, can oxidize thiol groups in the virus protein disulfide isomerase, rendering it unable to penetrate the healthy cell membrane. I'll repeat that, unable to penetrate the healthy cell membrane. In this way, selenite inhibits the entrance of viruses into the healthy cells and abolish their infectivity. Wow, inhibits the entrance of viruses into the healthy cells and abolish their infectivity. Therefore, this simple chemical compound can potentially be used in the recent battle against coronavirus epidemic, unquote. Where do I get my selenium from? Well, Brazil nuts are the richest source of selenium, and 4 to 6 Brazil nuts a day is what I have to get my selenium. And I also take some mineral supplements, usually shilajit or marine phytoplankton, where I also make sure that I'm getting my full spectrum of minerals. I also try and shop at farmers markets, talk to the farmer, try and find biodynamic farmers who are passionate about replenishing their soils with minerals. And I also grow a lot of my own food, and I use worm juice from my worm farm for the soil in the food that I'm growing, and I also apply ocean water, diluted. I apply ocean water and then I just hose it in to remineralize the soil with ocean minerals. A lot of really good books written about the use of ocean minerals for plants, and we know from the fact that when I think it was the giant flood in Thailand where the tidal wave washed over the rice fields and everybody thought the rice would be ruined for the next few years, and in fact they had bumper crops because the soil was remineralized and they allowed the rice to grow better. Right, next one, Journal of Neurotoxicology, talking about phthalate exposure. Phthalate is part of plastics in pregnant women, and if that has any effects on the neurodevelopment of their children, and it finds it does. Paper says prenatal phthalate chemicals may have adverse effects on brain development by various mechanisms, including oxidant damage. However, birth cohort findings have been conflicting. This study aimed to investigate the interplay between maternal prenatal phthalate levels, infant genetic vulnerability to oxidative stress, and child neurodevelopment. Higher prenatal phthalate levels and a higher oxidative stress genetic score were each associated with subsequent autism spectrum disorder. Consistent patterns were evident across gene score phthalate combinations for cognition, autism spectrum disorder, autism traits, and inattention/hyperactivity. Other putative oxidant factors such as prenatal smoking further increase risk. Prenatal phthalate levels and infant oxidative stress-related genetic vulnerability are associated with adverse neurodevelopment. Combined exposures are important. Current recommendations and regulation on maternal phthalate exposure during pregnancy require re-evaluation. To summarise that research was done in Victoria, Australia, and down south of Australia. Summarise, it's pretty straightforward. It says that plastic exposure to pregnant women increases the risk of autism. You've got to remember there's plastic inside coffee cups, so I've completely given up paper coffee cups with their plastic, and don't drink anything out of a plastic water bottle because of the phthalate and the bisphenol A in some of the bottles. But exposure to those phthalates was increasing the risk of attention deficit hyperactivity disorder and autism, and that was even worse if there was smoking. So if I was a pregnant woman, I certainly wouldn't be smoking, and I certainly would drastically reduce my exposure to plastics, particularly from plastic water bottles and from paper coffee cups. I think the whole world now is moving to, you know, taking along their own cups to coffee shops as well, and I'm always I've got a metal thermos that I take with me that I now use. Right, the next one is published in Science Direct. It's called Inflammatory Bowel Diseases, the Hygiene Hypothesis and the Other Side of the Microbiome, Parasites and Fungi. And what this review says is it tackles the concept of the evolutionary mismatch in relation with the reduction of the prevalence of the so-called dirty old friends. The dirty old friends. Never thought I'd read that in a medical journal. These formed the variegated community of parasites and microorganisms, either prokaryotic or eukaryotic, that over long evolutionary times co-evolved with humans and their ancestors, inhabiting their digestive tracts and other body districts. This community of microbial symbionts and metazoan parasites is thought to have evolved a complex network of interdependence with the host, in particular in relation with their immune stimulating capacity and with the consequent adaptation of the host immune response to this chronic stimulation. Strictly related to this evolutionary mismatch, the hygiene hypothesis proposed by David Strawn in 1989 foresees that the increase in the incidence of inflammatory and autoimmune disorders during the 20th century has been caused by the reduced exposure to parasites and microorganisms, especially in an industrialized country. So what they're doing is they're extrapolating the hygiene hypothesis that why our immune systems aren't as strong as they used to is because we're not getting exposed to the same sort of microbes in our environment, because we're not in the garden with our hands in the soil and we're cleaning everything and we're washing everything, and that's harming our immune system because it's not getting stimulated by those microbes. And they're making it the case for parasites and fungi in the gut doing the same thing. And I think looking at other species, it would suggest that's the case. And looking at who has the healthiest microbiomes in the world, which is the hunters and gatherers of Tanzania, that would suggest that we are these species are interdependent. They need us, we need them. We don't need them, but we need the fight. It's the concept of hormesis, that a short-term stress, fighting a short-term stress and recovering from it is good for us. A chronic stress is a disaster, but short-term stress. And if you look at all the other animal species that we've studied who get affected by parasites, the ones we've studied always have a defense system against them. It doesn't matter if you're a chimpanzee which changes their diet, or some insects which have strategies for when they get parasites. It's the fight which makes us stronger. Going to the gym for 30 minutes and exercising, even though it hurts at the time, makes us stronger. So having parasites, having these fungi, they're saying it's the fight by getting rid of them which makes us stronger and gives us a stronger microbiome. Next, melanopsin-dependent photoreception provides earliest light detection in the mammalian retina. This is an article from a few years ago which I just found, which talks about the development of the eye. And it says, the visual system is now known to be composed of image-forming and non-image-forming pathways. Photoreception for the image-forming pathway begins at the rods and cones, whereas that for the non-image-forming pathway also involves intrinsically photosensitive retinal ganglion cells which express the photopigment melanopsin. melanopsin in our eye and our skin is responsible for the detection of light rather than the seeing of objects. The findings suggest that the non-image forming pathway is functional long before the mainstream image forming pathway during development. That tells us how important light is to us as a species, that it develops way before our ability to actually recognize objects, and that's through these melanopsin receptors in our skin and our eye and the detection of light, which is why there's so much research now about the circadian rhythm and the different frequencies of light that we're getting exposed to throughout the day. Can selenium combat cancer? World Health Net, see what it says. A study published in the Journal of Cancer Prevention showed that selenium helped to repair DNA damage that was caused by carcinogens, and revealed that selenium also helped to stop cell migration and invasion, particularly in breast, colon, melanoma, liver, lung, and prostate cancers. Carcinogens induce genetic damage when they latch onto DNA and alter its components. To combat this, anti-carcinogens neutralize enzymes that enable carcinogens to damage DNA. So anti-carcinogens are stopping these carcinogens damaging our DNA. Selenium also helps to prevent further DNA damage and helps to improve DNA repair capacity by boosting the protective activities of DNA repair enzymes. So not only does selenium help neutralize the carcinogens, it helps to repair any damage to the DNA caused by those carcinogens. This study suggests that DNA repair capacity can also be enhanced when selenium is paired with zinc, which boosts the antioxidant and chemoprotective activities of selenium while delivering its own immune function boosting properties. Well, we know that zinc and selenium pair up with our immune system. Selenium stops the virus from attaching to our cells and injecting its DNA, and zinc helps kill the virus directly and through metallothioneins. So not only does zinc and selenium pair up in our immune system, also appears that they pair up in our ability to prevent cancer. There is no consensus among experts that selenium can be used as a treatment for cancer, but extensive studies provide compelling evidence that it can help to significantly reduce the risk of certain cancers and minimize the effects of cancer on the body. Zinc and selenium, immune system and cancer. Let's have a look at two new studies on vitamin D. One was a quick one which showed that low levels of vitamin D increase the risk of incontinence in women. So important for women again to get outside and get some sun to minimize their risk of incontinence. But here was the big one. This is a study which is May 2020 in Toxicology Applied Pharmacology. Vitamin D supplementation rescues statin-induced myopathy in mice via improving mitochondrial crystal shape. And it says mitochondrial homeostasis plays a role in the process of energy metabolism, i.e. mitochondria produce our energy for our body in the form of ATP. Thus, the mitochondrial dysfunction is a mortal damage, more expressive language in medical studies. Thus, the mitochondrial dysfunction is a mortal damage for the highly energy requiring tissue. In our study, the mitochondrial cristae observed under transmission electron microscope. If you search for mitochondria on the internet and look at images, you'll see pictures of the mitochondria, and you'll notice that the inner membrane of the mitochondria has these projections, which is like finger, coming in from the inner mitochondrial membrane, and that's where a lot of the energy production takes place. So when I say cristae, I'm talking about these inner projections of the mitochondrial membrane. In our study, the mitochondrial cristae observed under electron microscopy were lytic in statin-treated gastrocnemius. That means the statin drugs were damaging the inner membrane of the mitochondria. Interestingly, vitamin D supplementation improved the mitochondrial cristae shape. As expected, the mitochondrial dysfunction and oxidative stress was mitigated by vitamin D supplementation. In conclusion, these findings suggested that moderate vitamin D supplementation rescued statin-induced myopathy, myopathy is muscle pain caused by the statins, by improving the mitochondrial cristae shape and function. So I'm not on statins, but if I was on a statin, I would be out getting some sun to keep my vitamin D levels up. And finally, in this month's International Journal of Mycobacteriology, we've got a study on the spectrum of pulmonary fungal pathogens, associated risk factors, and antifungal susceptibility pattern among persons with presumptive tuberculosis. So in this study, they took over 200 people in Nigeria who they thought had pulmonary lung tuberculosis. And then they checked them for fungus as well. Now this is much more detailed checking for fungus, because as you'll see that they describe in the study, it's actually quite difficult to diagnose a fungal infection in the lungs. So they did more dedicated testing to try and identify how many of these people who they thought had pulmonary tuberculosis had tuberculosis versus having fungus. And this is what they found. Pulmonary mycosis, that's fungal infections of the lungs, poses a great diagnostic challenge because it's hard to diagnose fungus. Due to the lack of pathognomonic and radiological features, we don't have good blood tests, we don't have good X-ray appearances to tell us that it's fungus. Especially in the absence of mycology lab tests, this study was aimed to isolate, phenotypically identify, determine the prevalence of pulmonary fungal pathogens. And what they found was that of the 216 people, 20% of them turned out to have tuberculosis, and 74% had pulmonary fungal pathogens. Among the isolated pulmonary fungal pathogens, Aspergillus fumigatus made the highest occurrence, while 7% had other fungi there as well. Conclusion: findings from this study revealed high-level pulmonary fungal pathogens, especially among people who were thought to have tuberculosis. A majority of fungal isolates were resistant to fluconazole, that's diflucan, an antifungal medication. It's recommended that persons should do away with or minimize risk factors for pulmonary fungal pathogens identified in the study. And of course, our biggest risk for pulmonary fungal pathogens is living in a moldy house. So for the first thing that I do when I make sure that my risk of getting a fungal infection is decreased is I make sure that my house has no moisture or minimal moisture in it, and you can buy moisture meters at hardware shops to check the moisture in the walls to minimize the risk of my house developing mold. If you've enjoyed today's session, make sure you give the podcast a review, good one hopefully, and like the video and share it on YouTube. You can also support me on my Patreon page, where I go into much more depth about these studies and what I'm doing to mitigate or introduce into my lifestyle, my program to stay maximally healthy, optimize my performance, and live for the longest period of time with the maximum amount of health. Then I go into much more detail on my Patreon page, which is www.patreon.com/Dr. Greg Emerson. See you all next week. Thanks for watching.